does not arise. This technique is very simple and healthy seed can be produced. Fish can be spawned more than once in 1 year. Hybridization is possible, and in induced breeding techniques, four main types of materials are used to give injections to fish: pituitary gland extractions, HCG, ovaprim, and ovatide. Induced breeding with pituitary gland extractions. Fish breeding by pituitary gland extraction is an effective and dependable way of obtaining pure seed of cultivable fishes and is practiced today on a fairly extensive scale in India as well as many other countries in the world. It involves injecting mature female and male fishes with extracts of pituitary glands taken from other mature fish. Fish pituitary gland. Fish pituitary gland is a small, soft body and creamy white in color. It is more or less round in carbs. It lies on the ventral side of the brain behind the optic chiasma in a concavity of the floor of the brain box known as cella tarsica, and enclosed by a thin membrane called dura mater. In few fishes, it is attached to the brain by a thin stalk known as the infundibular stalk. Based on the infundibular stalk, the glands are classified into two types, namely platybasic, leptobasic. Platybasic means it is without stalk and it has an open infundibular recess. Leptobasic means it is with stalk, have obliteral infundibular recess. Leptobasic type of pituitary glands are found in carps and platybasic type found in Chanidae and Nannidae. The size and weight of the gland varies according to the size and weight of the fish. In Labiorhita, the average weight of the pituitary gland is 6.6 mg in 1 to 2 kgs fish and 10.3 mg in 2 to 3 kg fish and 15.2 milligrams in 3 to 4 kg fish and 18.6 mg in 4 to 5 kg fish. Pituitary gland secretes the gonadotrophic hormones, FSH, or follicular stimulating hormone, and LH, which is also called as luteinizing hormone. Both hormones are secreted throughout the year, but the proportion in which they are secreted is directly correlated with the cycle of gonadal maturity. The FSH causes the growth and maturation of ovarian follicles in females and spermatogenesis in the testes of males. Luteinizing hormone helps in transforming the ovarian follicles into corpus luteum in females and promoting the production of testosterone in males. These hormones are not species specific. That is, a hormone obtained from one species is capable of stimulating the gonads of another fish. However, there is great variability in its effectiveness in different species. Experiments conducted on induced breeding of fishes, they have clearly shown the relative effectiveness of fish pituitary extracts over mammalian pituitary hormones, sex hormones, and various steroids. This is the reason why fish pituitary is being extensively used today in fish breeding work all over the world. Collection of pituitary gland. So how to collect the gland from the fish? The fish from which the pituitary gland is collected is called the donor fish. The success in induced breeding of fish depends to a great extent on the proper selection of the donor fish. The gland should preferably be collected from fully ripe gravid fishes, as the gland is most potent at the time of breeding or just before spawning. The potency of the gland decreases after spawning. Glands collected from immature or spent fishes usually do not give satisfactory results. Glands in induced breed fishes collected immediately after spawning have also been found to be effective and can be used for breeding of other fishes. Most suitable time in India for collection of pituitary glands of major carbs is during May to July months, as the majority of carbs attain advanced stages of their maturity during this period. Since common carp, Cyprinus carpio, is a perennial breeder, its mature individual can be obtained almost all the year round for the collection of glands. The glands are usually preferred to be collected from freshly killed fishes, but those collected from ice preserved specimens are also used. In India, the commonly adopted technique of gland collection is by chopping off the scalp of the fish skull by an oblique stroke of a butcher's knife. After the scalp is removed, the gray matter and fatty substances lying over the brain are gently cleaned with a piece of cotton. The brain, thus exposed, is carefully lifted out by detaching it from the nerves. In majority of the cyprinids, when the brain is lifted, the gland is left behind on the floor of the brain box. The dura mater covering the gland is then cautiously removed using a fine needle and forceps. The exposed gland is then picked up intact without causing any damage to it because damaged and broken glands result in loss of potency. Glands are also collected through foramen magnum. It is in fact a much easier method of gland removal, which is commonly practiced by the professionals for mass scale collection in crowded and noisy fish markets. In this method of gland collection, the fish is required to be essentially beheaded. In markets, glands are collected from fish heads that are already cut by retailers. In the cut fish heads, the foramen can be clearly seen from behind, holding gray matter and fatty substances in it. The brain lies on the ventral sides of the foramen. For taking out the gland, the gray matter and fatty substances are first removed by inserting the blunt end of the forceps into the foramen and pulling out the entire matter without disturbing the brain. The brain is lifted up carefully and pushed forward or is pulled out of the hole. The gland lying at the floor of the brain box is then picked up using a pair of the fine tweezers. An experimental worker easily manages to collect about 50 to 60 glands in 1 hour by adopting this technique of collection. Preservation of pituitary glands. If the collected glands are not meant for use then and there, they must be preserved. Due to their glyco or mucoprotein nature, they are liable to immediate enzymatic action. The pituitary glands can be preserved by three methods: absolute alcohol, acetone, and freezing. Preservation of fish pituitary gland in absolute alcohol is preferred in India. Moreover, experiments done so far with alcohol preserved glands on Indian major carbs have given more positive results than with acetone preserved glands. The gland after collection is immediately kept in absolute alcohol for defatting and dehydration. Each gland is kept in a separate fuel marked serially to facilitate identification. After 24 hours, the glands are washed with absolute alcohol and kept again in fresh absolute alcohol contained in dark color bottles and stored either at room temperature or in a refrigerator. Occasional changing of alcohol helps in keeping the glands in good condition for longer periods. In order to prevent moisture from getting inside the fuels, they may be kept inside a desiccator containing some anhydrous calcium chloride. It is preferable to keep the glands in a refrigerator. They can be stored in a refrigerator up to 2 to 3 years and at room temperature up to 1 year. Acetone also is a good preservative. In this method, soon after collection, the glands are kept in fresh acetone or in dry ice chilled acetone inside a refrigerator at 10 degrees centigrade for 36 to 48 hours. During this period, the acetone is changed 2 to 3 times at about 8 to 12 hours intervals for proper defatting and dehydration. The glands are then taken out of acetone, put on a filter paper, and allowed to dry at room temperature for 1 hour. They are then stored in a refrigerator at 10 degrees centigrade, preferably in a desiccator charged with calcium chloride or any other drying agents. The preservation of glands in acetone is largely practiced in Russia and America. Preparation of pituitary gland extract. Preserved glands are then weighed. This is essential for accurate determination of the dose to be given according to the weight of the breeders. The weight of the gland may be taken individually or in a group. To get a more accurate weight, a gland should be weighed exactly after 2 minutes of its removal from alcohol. The pituitary extract should be prepared just before the time of injection. The quantity of gland required for injection is at first calculated from the weight for the breeder to be injected. The glands are then selected and the required quantity of glands is taken out of the fields. The alcohol is allowed to evaporate. If the glands are alcohol preserved ones, acetone dried glands are straight away taken from the fields for maceration. The glands are then macerated in a tissue homogenizer by adding a measured quantity of distilled water or common salt solution or any physiological solution which is isotonic with the blood of the recipient fish. The most successful results of induced breeding in the Indian major carps have so far been obtained with distilled water and 0.3% common salt solution. The concentration of the extract is usually kept in the range of 1 to 4 milligram of gland per 0 to 1 ml of the media. That is, at the rate of 20 to 30 grams of the gland in 1 ml of the media. After homogenization, the suspension is transferred into a centrifuge tube. While transferring the homogenate, it should be shaken well so that settled down gland particles being mixed with the solution come into the centrifuge tube. The extract in the tube is centrifuged and the supernatant fluid is drawn into a hypodermic syringe for injection. The pituitary extract can also be prepared in bulk and preserved in glycerin. 1 part of extract proportionate to 2 parts of glycerin. Before the fish breeding season, so that the botheration of preparing extract every time before injection is avoided. The stock extract should always be stored in a refrigerator or in ice. Technique of breeding. The induced breeding operation of major carps is taken up when regular monsoon sets in the fishes become fully ripe and water temperature goes down. Females having a round, soft and bulging abdomen with swollen reddish vent and males with freely oozing milt are selected for breeding. A male breeder can also be easily distinguished by roughness on the dorsal surface of its pectoral fins. The most important aspect of induced breeding of fish is the assessment of proper dosages of pituitary extract. The potency of the gland varies according to the size and stages of sexual development of the donor, as well as the species of the donor fish, time of collection of glands, and their proper preservation. The dose of the pituitary gland is calculated in relation to the weight of the breeders to be injected. It has also been noticed that identical doses to breeders of similar weights may give contradictory results owing to difference in maturity of gonads. So even heavy doses of hormones may not be effective if the gonads are in the resorption stage. By careful selection of breeders and administering a known weight of pituitary gland extract per kg body weight of the breeders, successful breeding can be obtained. Experiments on standardization of doses indicate that administration of a preliminary low dose in the female breeder followed by a higher effective dose after 6 hours proves more successful than a single knockout dose. A single high dose has been found useful when the breeders are in ideal condition and the weather is favorable. Rohu responds well to 2 injections, while Katla and Mrigal to both 1 and 2 injections. An initial dose at the rate of 2 to 3 mg of pituitary gland per cage body weight of fish is administered to the female breeder. On male breeders do not require any initial dose. If they ooze milt on slight pressure on their abdomen, 2 males against each female make a breeding set. To make a good matching set, the weight of the males together should be equal or more than the female. In case the condition of any one of the 2 males is not found in the freely oozing stage, an initial injection may be administered to the male at the rate of 2 to 3 mg per kg body weight. After 6 hours, a second dose of 5 to 8 milligram per kg body weight is given to the female, while both the males receive the first or second dose at the rate of 2 to 3 mg per kg body weight. Slight alterations in doses may be made depending upon the condition of maturity of the breeders and the prevailing environmental factors. In the absence of a chemical balance, 1, 2, 3 pituitary glands are effective for a pair of fish. Now, how to inject the injections? Intracranial injections are preferred in USSR and intraperitoneal in USA and Japan. Intramuscular injection is the most common practice in India. The intramuscular injection is less risky in comparison with the other methods. Intraperitoneal injections are usually given through the soft regions of the body, generally at the base of the pelvic fin or sometimes at the base of the pectoral fin. But there is some risk of damaging the internal organs, especially the distended gonads when administering an intraperitoneal injection in fully mature fishes. Injections are usually given at the caudal peduncle or shoulder regions near the base of the dorsal fin. While giving injections to the carps, the needle is inserted under a scale, keeping it parallel to the body of the fish at first and then pierced into the muscle at an angle. There is no hard and fast rule regarding the time of injection. Injections can be given at any time of the day and night. But since low temperature is helpful and the nighttime remains comparatively quieter, the injections are generally given in the late afternoon or evening hours with timings so adjusted that the fish is able to use the quietude of the night for undisturbed spawning. The most convenient hypodermic syringe used for the purpose is a 2 cc syringe having graduations of 0.1 cc division. The size of the needle for the syringe depends upon the size of the breeders to be injected. Number 22 needle is conveniently used for 1 to 3 kg carps. Number 19 for larger carps. And number 24 can be used for smaller carps. Use of anesthetics during injection would significantly increase the survival of brood fish. Commonly used anesthetics are MS-222 and quinaldine. MS-222 may be added to water in doses of 50 to 100 mg per liter. A roll of cotton soaked in a 0.04 m of this solution can be inserted into the mouth of the fish. Quinaldine is used at the rate of 50 to 100 mg per liter. You can see in the picture how the sperm is extracted. from the fish and how the steroids or the injections are given to the fish. Next, coming to breeding harpa and spawning. After the injection, the breeders are released immediately inside the breeding harpa. The breeding harpa is generally made of fine cloth in the size of 3.5 to 3.5 into 1.5 into 1 meter for larger breeders and 2.5 into 1.2 into 1 meter for breeders weighing less than 3 kg. All the sides of the breeding harpa are stitched and closed, excepting a portion at the top for introducing the breeders inside. Generally, one set of breeders is released inside each breeding harpa. But sometimes, in order to save on pituitary material, community breeding is also tried by reducing the number of male breeders. After the release of the fish, the opening of the harpa is securely closed so that breeders may not jump out and escape. Instead of harpas, cement cisterns or plastic pools as big as harpas can also be used for breeding. Spawning normally occurs within 3 to 6 hours after the second injection. Soon after fertilization, the eggs swell up considerably owing to absorption of water. Fertilized eggs of major carps appear like shining glass heads of crystal clear transparency, while the unfertilized ones look opaque and whitish. The size of eggs from the same species of different breeders varies considerably. Fully swollen eggs of the Indian major carps measure 2.5 mm in diameter, the largest being that of Katla and the smallest of Rohu. The carp eggs are non-floating and non-adhesive type. The yolk possesses no oil globule. The Indian major carps have a profuse egg-laying capacity. Their fecundity on an average is 3.1 lakh in Rohu, 1 to 3 lakh in Katla, and 1.5 lakh in Mrigal. The developing eggs are retained in the breeding harpa undisturbed for a period of at least 4 to 5 hours after spawning to allow the eggs to get properly water hardened. After this, the eggs are collected from the harpa using a mug and transferred into a bucket with a small amount of water. The breeders are then taken out and weighed to find out the difference before and after spawning. This gives an idea of the quantity of the eggs laid. The total volume and number of eggs can be easily calculated from the known volume and the number of eggs of the sample mug. Percentage of fertilized eggs is also assessed accordingly by conducting random sampling before and after spawning. This gives an idea of the quantity of the eggs laid. The total volume and number of eggs can be easily calculated from the known volume and the number of eggs of the sample mug. Percentage of fertilized eggs is also assessed accordingly by carrying out random sampling. Then coming to stripping. Chinese carps, however, do not spawn naturally. And when they spawn, the percentage of fertilization is generally very low. Stripping or artificial insemination is therefore followed. The female fish is held with its head slanting upwards and tail down and belly facing the vessel. And the eggs are collected into an enamel or plastic trough by pressing the body of the female. The male fish is then similarly held and milk is squeezed out into the same trough. The gametes are then mixed as soon as possible by means of a quill feather to allow fertilization. The fertilized eggs are then washed a few times with clean water to remove excess milk and allowed to stay undisturbed in fresh water for about 30 minutes. The eggs are then ready for release into the hatching tanks. Technique of hatching the eggs. The eggs collected from the breeding hapa are transferred into the hatching hapas. A hatching hapa consists of two separate pieces of hapas, the outer hapa and the inner hapa. The inner hapa is smaller in size and is fitted inside the outer hapa. The outer hapa is made up of a thin cloth in the standard size of 2 into 1 into 1 meter, while the inner hapa is made of round meshed mosquito net cloth in the dimensions of 1.75 into 0.75 into 0.5 meters. All the corners of the outer and inner hapas are provided with loops and ropes to facilitate installation. About 75,000 to 1 lakh eggs are uniformly spread inside each inner hapa. The eggs hatch out in 14 to 20 hours at a temperature range from 24 to 31 degrees centigrade. The period of incubation, in fact, is inversely proportional to the temperature. After hatching, the hatchlings escape into the outer hapa through the meshes of the inner hapa. The inner hapa containing the eggshells and the dead eggs are removed when the hatching is complete. The, the hatchlings remain in outer hapa undisturbed till the third day after hatching. During this period, they subsist on the food stored up in their yolk sac. By the third day, the mouth is formed and the hatchlings begin directive movement and feeding. At this stage, they are carefully collected from the outer hatching hapa and stocked into prepared nurseries. Induced breeding with HCG. Today, pituitary gland extraction is a well-established technique for induced breeding all over the world. Its large-scale use poses the following problems with regard to availability and quality of pituitary gland. Inadequate supply of pituitary gland, high cost, variability in pituitary gonadotrophin potency, and cheating by unscrupulous PG suppliers. To overcome these problems, human chorionic gonadotrophin has been found as an alternative for pituitary gland. HCG was discovered in beginning of 1927 by Aschim and Zondek. They extracted good quality hormone with luteinizing gonadotrophic activity from the urine of pregnant women. HCG is a glycoprotein or CLO protein because of the carbohydrate molecules attached to the protein molecules. Its primary function is to maintain the production of estrogen and progesterone by the corpus luteum. It is produced by the placenta and excreted through the urine during early stages of pregnancy. HCG comprises of two subunits, A, and has a molecular size of 45,000 to 50,000 Daltons. There are 17 amino acids in it, out of which alanine, proline, serine, cysteine, and histidine are important. Due to the large number of amino acids, HCG has a high protein content. The molecular weight has been reported as 59,000 by gel filtration and 47,000 by sedimentation equilibrium. During early stages of pregnancy, HCG is rich in the urine of pregnant women. Several methods are employed for the extraction of HCG. Aschim and Zondek used ethanol for precipitation. Katzman and Siana used different absorbents. Commercial crude HCG extraction is made with gel filtration. Follicle stimulating hormone and utilizing hormone of the pituitary play an important role in the normal reproduction of fish. That is, in promoting the development of gonads, growth, maturity, and spawning. HCG is more or less similar in character and function to FSH and LH. As pituitary gland is used for induced fish breeding, HCG can also be used for early ripening of gonads. Superiority of HCG over pituitary gland can be measured on the following grounds. Fish attains maturity faster with HCG. The span of the breeding season can be increased with HCG, which ensures better survival of spawn. It reduces the time gap between preparatory and final doses. HCG is more economical and has a long shelf life. HCG is easily available from a standard source, hence is more reliable. Periodical injections of HCG throughout the year ensure better health and increase in weight and gonadal development potency of HCG is known. Available in neat packets of known weights, no preservation is involved, cannot be spurious. HCG treated fishes can be used more than once for induced breeding in the same season. Mortality rate of hatchlings is negligible. Consumption of the drug is less during induced breeding. HCG can be used as growth hormone, and the absorption of eggs at the end of the breeding season is comparatively less by the administration of HCG. The crude HCG is in powder form and gray, white, or light yellow in color. It dissolves easily in water. The calculated quantity of crude HCG is taken into a tissue homogenizer and stirred for 5 to 10 minutes with measured distilled water. It is centrifuged for 3 to 5 minutes. The clear light yellowish supernatant liquid having the HCG hormones is taken and injected immediately. Any delay in use will result in the loss of the potency. In case of silver carp, use of HCG is found to be quite successful. The dosage is 4 to 6 mg per kg body weight of male and 6 to 8 milligrams per kg body weight of first dose, and after about 6 to 7 hours 10 to 12 mg per kg body weight of second dose for female, which gave good results. Use of only HCG in the breeding of Indian major carps has not given successful results so far. A combination of 60 to 80% HCG and 40 to 20% pituitary gland for Indian major carps and grass carps is successful. Fishes which are induced to breed with HCG alone are mullets, Cyprinus carpio, Lactariolurus punctatus, Oreochromis nilotica, Artisticus nohilis, Misgernus fossilis, Esox lucius, and Epinephelus tovina. Recent work shows that the combination of HCG and PG is more commendable than HCG or PG alone. More work needs to be done to standardize the dosage of HCG for induced breeding of major carps and Chinese carps. Induced breeding with ovaprim. Due to the problem of varying potency of pituitaries, alternatives were tried. Attempts have been made in various countries to use the analogs of luteinizing hormones, releasing hormones for induced breeding of fishes with varying degrees of success. However, the success achieved with LH-RH was not always consistent, apart from its higher dose requirement for induction of spawning. This epoch making investigation paved the way for developing simple and effective technology for induced breeding of most of the cultivable fishes. In a joint collaborative project funded by International Development Research Center, Canada, to Dr. Lin of China and Dr. Peter of Canada, a series of investigations were carried out to develop a reliable technology for breeding fishes. Their investigations led to the development of a new technique called as LNPE method, wherein an analog of LHRS-RH is combined with a dopamine antagonist. Based on the principle, Sindel Laboratories Limited, Canada, has manufactured a new drug called as Ovaprim. Ovaprim is a ready-to-use product, and the solution is stable at ambient temperature. It contains an analog of 20 micrograms of salmon gonadotrophin releasing hormone and a dopamine antagonist, domperidone at 10 mg per ml. The potency of Ovaprim is uniform and contains a GNR1, which is known to be 17 times more potent than LHRH. The dopamine antagonist, domperidone used in Ovaprim, is also reported to be better than other commonly used antagonist, pimozide. Ovaprim, being a ready-to-use product and one which does not require refrigerated storage, appears to be the most convenient and effective ovulating agent. This drug is administered to both female and male brood fish simultaneously in a single dose. Unlike pituitary extract, which is given in two split doses, this reduces not only the handling of brood fish but also helps in saving considerable amount of time and labor, which will add on to the cost of seed production. The spawning response in treated species is found to be superior to the pituitary extract injected species. The efficiency of Ovaprim for induced breeding of carps have given highly encouraging results in Katla, Rohu, Mrigal, silver carp, grass carp, big head, etc. The effective dose required for various species of carps is found to vary considerably. The common dose for all carps is 0.10 to 0.2 ml Ovaprim per kg body weight of males and 0.25 to 0.80 ml Ovaprim per kg body weight of females. Female Katla is found to respond positively for a dose range of between 0.4 to 0.5 ml per kg, while Rohu and Mrigal respond to lower doses of 0.35 ml per kg and 0.25 ml per kg respectively. Ovaprim has unique advantages over pituitary hormone as ready-to-use liquid form in 10 ml vial, consistent potency and reliable results, long shelf life, and can be stored at room temperature. Formulated to prevent overdosing, male and female can be injected only once simultaneously, reduces handling and post-breeding mortality. Repeated spawning possible later in the season and high percentage of eggs, fertilization and hatching. Then coming to induced breeding with overtide. Overtide is an indigenous, cost-effective and new hormonal formulation for induced breeding of fishes. The new formulation is having the base of a synthetic peptide, which is structurally related to the naturally occurring hormone, gonadotrophin releasing hormone. Gonadotrophin releasing hormone is not a steroidal hormone and belongs to the class of organic substances called peptides. It is presented as a low viscosity injectable solution, which is not only highly active but also cost effective compared to other commercially available spawning agents. It is also effective in breeding major carps and catfishes. The doses for females are 0.20 to 0.40 ml per kg for Rohu and Mrigal, 0.40 to 0.50 ml per kg for Katla, silver carp and grass carp, and 0.20 to 0.30 ml per kg for Kalbasul. The dosages for males are 0.10 to 0.20 ml per kg for Rohu, Mrigal and Kalbasu. 0.20 to 0.30 ml per kg for Katla and 0.20 to 0.25 ml for kg for silver carp and grass carp. The advantage of overtide are it is cost effective hormonal preparation. It gives high fertilization and hatching percentage, that is 85 to 95 percentage. It increases egg production through complete spawning, produces healthy seed. It is easy to inject due to its low viscosity. It does not cause adverse effects on brood fish after injection. It can be administered in a single dose of brooders. Then induced breeding with OOPL. OOPL developed by the University of Godola in Hungary. It is a preparation containing mammalian GnRH and the water-soluble dopamine receptor antagonist metoclo metoclopramide. The concentration of DALA6, Pro9NET, GnRH, and metoclopramide are in the form of 18 to 20 micrograms per pellets and 8 to 10 milligrams per pellets respectively. The hormone is thus available in pellet form. Each pellet contains superactive gonadotrophin releasing hypothalamic hormone analog with an equal effect which a 3 mg normal acetone dried dehydrated carb hypophysis gland has. Induced propagation of fish had been shown to be more effective if the hormone was administered in 2 doses. Prime dose and dissolving dose, as reported by Szabo in 1996. Cyprinid successful results were reported when 2 to 2.5 pellets per kg were administered to female broodfish. However, preliminary trials with single injection of OVPL gave encouraging result on a few species of Indian major carps and Clarius batrachus. The required amount of OVPL was calculated on the basis of weight and condition of broodfish. The pellets were pulverized in a mortar and dissolved in distilled water. The trials were conducted in July to August of 1999. Other substances used for induced breeding, like LHRS analogs, steroids, and clomiphene, are used for induced breeding of fishes. LHRH analog. Various analogs of luteinizing hormone, releasing hormone, have been used for induced breeding of fishes. Investigations have revealed that the potential action of releasing hormone when dopamine antagonist is simultaneously used with the analogs is used successfully in China. An analog of teleost GnRH is found to be more potent than LHRH. GnRH stimulates GTH, gonadotrophin hormone, in teleos, dosage 25 to 100 micrograms per kg. Steroids. Selected steroid hormones are used to induce fish. The effects of steroid hormones on ovulation are seen primarily as germinal vesicle breakdown. Ovulated oocytes require at least 4 hours to become fertilized in mullets, whereas in most of the fishes, oocytes are fertilized immediately. The action of pituitary gonadotrophins on oocyte maturation is known to be mediated through steroid hormones. Deoxycorticosterone acetate and cortisone effectively stimulate ovulation in heteroneustous fossils. 17A-hydroxy-20B dehydroprogesterone is useful to induce goldfish, trout, and pikes. Other steroid hormones commonly used for spawning teleos are cortisone acetate, deoxycortisol, etc. Breeding of common carp. Common carp generally breeds in confined water. Spawning takes place in shallow marginal weed infected areas from January to March and from July to August. Common carp is also observed to breed round the year. Controlled breeding of common carp is conducted to achieve better spawning and hatching. A set of selected brooders, one female and two males, are put together in breeding harpa. In order to ensure successful spawning, sometimes the female fish is injected with pituitary gland extract at a low dose of 2 to 3 mg per kg body weight. Freshly washed aquatic weeds, example hydrilla, najas, eichhornia, etc., uniformly distributed inside the hapa. These aquatic weeds act as egg collections. The quantity of weed used in roughly double the weight of the female introduced. Each weed attached with 40,000 to 1 lakh eggs are distributed into a single hatching hapa. After 4 or 5 days, the weeds are taken out carefully. Then estimation of eggs. The eggs are collected from the hapa by means of cup or tray or beaker and transferred to the buckets. The breeders are also removed from the hapa and their weights are noted. The difference in weights reveals approximately the number of eggs laid. The eggs are kept in a rectangular piece of mosquito net and allow the water to drain. allow the water. Then percentage of fertilization can be arrived at by counting the number of fertilized eggs from egg samples of 1 ml measure. Now, before going to model examination questions, let us look into the summary. Fish seed is obtained from 3 sources: riverine, hatcheries, and bunds. Collection of seed from riverine source is an aged practices. Hatcheries are the best way of getting fish seed. Carps breeds in flowing water like rivers. Induced breeding is an excellent technique to get pure and required fish seed. In induced breeding techniques, four main types of materials are used: pituitary gland, extraction, HCG, ovaprims, and ovatide. Several techniques are adapted for the collection of pituitary gland in different countries. The pituitary glands can be preserved by the 3 methods of absolute alcohol, acetone, and freezing. The induced breeding operations of major carps is taken up when regular monsoon sets in. By careful selection of breeders and administrating a known weight of pituitary gland extract per kg body weight of the breeders, successful breeding can be obtained. 2 males against each female make a breeding set. Pituitary injections are usually given at the caudal peduncle or shoulder region near the base of the dorsal fin. After the injection, breeders are released immediately inside the breeding hapa. Spawning occurs within 3 to 6 hours after the second injection. Chinese calves do not spawn naturally. Stripping or artificial insemination is therefore followed. The eggs collected from breeding hapas are transferred into the hatching hapas. Indian major calves could be induced to spawn twice the same season with an interval of 2 months. Human chorionic gonadotrophin, that is HCG, has been found as an alternative for pituitary gland. Ovaprim is ready to use product and the solution is stable at ambient temperature. Ovatide is an indigenous, cost-effective, and new hormonal formulation for induced breeding of fishes. Ovapel is another hormonal product which is available in pellet form. Another substance like LHRH, analogs, steroids, and clomiphene are used for induced breeding fishes. The eggs are measured in a beaker, mug, or a cup of known volume and transferred to hatcheries. In the case of grass carp and silver crab, the induced breeding is supplemented either by dry stripping or wet stripping. Common crab breeds in confined water round the year. Now, if you see the model examination questions, these are the questions which are frequently asked. Answer the following in about 30 lines each. Write about fish pituitary gland, its collection. Write about preservation of pituitary glands and preservation of extract. Preparation of pituitary gland extract. Write about doses of pituitary extract. Write about breeding harpa, spawning and stripping. Write about induced breeding with HCG. Write about induced breeding with ovaprim. Write about induced breeding with ovatide. And write about other substances used for induced breeding. With this, we have come to the end of the chapter. Hope you all have understood. Thank you.